Hi everyone. This presentation is about vulnerable marine ecosystem, VMEs, and an overview of promising methodologies that could be employed to model their distribution in the Southern Ocean. First, an introduction of VMEs. VMEs have been defined as habitat of high functional significance while being fragile to any disturbance. More specifically, the FAO established 5 criteria to identify an habitat as VME, such as uniqueness, fragility, or structural complexity, and provided some example of such ecosystem as general guidelines, such as Simmons, sponge gardens, but also indicator taxa, such as CPENS. However, the presence of one of these indicator taxa is not sufficient to identify a VME. Another important point is no specific quantitative definition of a VME has been provided by the FAO because vulnerability is a continuum among species and because vulnerability is related to a type of and proportion of threat. Therefore, we will see that when modeling VMEs, it is important to have a clear quantitative definition of vulnerability and of VMEs for a particular application and for a particular threat. VME protection is a worldwide movement. In particular, it has been prompted by the UNGA resolution in 2006, which urged regional fisheries management organization and arrangement to protect VMEs. RFMOs implemented different conservation measures, such as MPAs and VMEs encounter thresholds. This can be found in the FAO VME database, as shown on the right image, a view of the North Atlantic VME protection. Despite these important efforts, according to the recent reviews, VME protection remains overall insufficient. Before looking at why that's insufficient, let's look more in detail to VMEs protection policy in the Southern Ocean. So in the Southern Ocean, VMEs are under the responsibility of CAMELA, which really implemented a precautionary approach to protect VMEs compared to other RFMOs, both in terms of spatial closures and data collection. CAMELA also provided some examples of VMEs and indicator taxa. This list is specific to the Southern Ocean. Also note that these taxa have been identified based on criteria slightly different from the FAO criteria, in particular the two greens criteria, so the larval dispersal and the motility ability. CAMELA is really encouraging the data collection for VMEs and reports it on its GIS portal. That's what is represented on the map. What we can observe, however, is VME data spareness, mostly concentrated in fishing areas or more often surveyed by scientific boats. CAMELA also made mandatory for the fishing boats to report the incidental take of VMEs indicator taxa and makes the record available. We can see the occurrence varies across taxa. We have a lot of sponges record, not much of ascidians, which is not easy to interpret since this record reflects in part an effect of catchability rather than the fishing impact, and in addition to the natural species abundance and distribution variability. So these two aspects, data spareness and type of data we have, make our knowledge of VMEs in the Southern Ocean very limited. Underwater imagery could definitely help, but it's very expensive and time consuming. But there is a solution that could bridge this gap between data spareness and limited knowledge of spatial distribution. That's species distribution modeling. That's what we are going to talk about now. Distribution modeling could indeed be very interesting since it could predict potential distribution of VMEs beyond sampled areas and provide some insight on their environmental requirements or inform science-based management. VME distribution have been used in other oceans for mainly 6 applications, 6 aims. In the next slides, I'm going to detail methodologies related to 3 of them, here in blue, which I think are of particular interest for the Southern Ocean. First aim, the reserve assessment. So the point here is, depending on the type of data you got, so presence only or presence absence data, the estimated quantity is not the same, which I like the importance of having a data type that fits your application. So 2 cases, either you have presence only data, often from incidental take of a taxa, let's say a sponge taxon, or you have a presence absence or abundance data from imagery data at final resolution, and you can define an habitat based on a certain density of species. In both cases, you will run your model, have some predictions, and make a comparison with the existing reserve network. But the quantities that you can estimate depending on your data type is different, which is important to have in mind. What do these numbers mean before redesigning your reserve network? Another application is the identification of critical areas for a particular threat. So let's say the fishing. Here we can have either presence absence or presence only or both. Do some prediction, complete the mapping of vulnerability for fishing threat, compare it to the fishing pressure to identify sites which within a certain risk. The third application is the identification of biodiversity hotspots, some predictions. However, here again, the estimated quantities vary depending on the type of data you got as input, and some site ranking can be done to identify biodiversity hotspots. So to summarize, VME's distribution has not been modeled yet around the entire Antarctica. And I think there are 3 main challenges for this. The lack of presence, absence, or abundance data, the lack of quantitative and qualitative definition of VMEs. So what density of sponges is needed to identify a sponge garden as VME, and which taxa assemblages or communities are considered as indicative of VMEs in the Southern Ocean. And to answer this question, a first good thing to do would be to look at what has been done in other oceans. I put down some interesting references of density-based definition and VME's community characterization. I now would like to talk about where I think the future of VME's distribution modeling is in the Southern Ocean, so some promising avenues. First, the environmental data. I highly recommend you to listen to Jansen's talk about fine resolution environmental predictors in the Southern Ocean. Concerning the biological data, there is a very important work done by Victor and colleagues to put together a very large and open source database of biological data based on imagery. And to have abundance data opens the door to more informative modeling strategies than presence only, such as joint species distribution, Bayesian approaches, and convolutional neural networks. Regarding the final output, I said earlier, the importance of assessing vulnerability to a particular type of threat, and the importance of reporting the uncertainty of your modeling to inform conservation management decisions. To conclude, VME's distribution modeling in the Southern Ocean needs a more specific and quantitative definition of VMEs, so density-based and community characterization. Two, we saw the importance to pay a special attention whether our data type, so presence only or presence absence, fits our application, as well as the way we assemble taxa predictions. Lastly, the emergence of new and very informative open source databases opened the door to more informative and reliable modeling approaches that could fit conservation management. That's the end of this presentation. Thanks for watching and see you around.